so I'm going to do this a little bit different from the previous talks. I see if I could actually get you on my computer screen and show you in real time what's actually happening in my group. So I'm at the medical school, and my research group does basically 3 different kinds of things. We do clinical trials. I'm a practicing physician as well in the breast center. We also do translational research, which mostly involves analyzing human tissues and trying to learn from them, learn how we could better predict disease outcome or what treatment would work best for a particular individual, and also to identify new therapeutic targets. And the third component is really laboratory research. So I'm going to walk you through, this is our web page. I mean, it's actually very easy to find out what people do just by simply Google searching their name and their publications. And this is the actual website, the internal year website that you can easily access. So I'm not going to talk much about the clinical faculty, but this is the research faculty. So Kim is an associate research scientist with a PhD in immunology. Christos is a computational biologist and adjunct professor now with us. And Mikhail is also a visiting scientist who retains a position with us to help with computational biology analysis. And Vinesh is also a vet lab PhD scientist. So this is the current trio of people who actually move in and move out. So these are visiting scientists and students. We work basically with 3 kinds of students. So my lab is rather diverse. Julia is a medical oncology fellow. So she's going to be a medical oncologist, an academic physician. She's currently finishing her medical training and does research in her final year. Hussein is a PhD student in your group in computational biology. Maria is also a medical oncology fellow at an earlier stage of her career. Yu is a postdoc working on developing a new antibody that we could possibly use as therapy. Tao is a visiting postdoc from Shanghai, and Xiaotong is a graduating PhD student. So, and there's another page here which is about past students. So that's another good way to learn about a laboratory, is that you actually look up what happened to the previous students. And so these are the previous students. So what we actually do, you can find on the list of publications. And it's a pretty good idea. It gives you a very good idea what people do by simply looking at what they published. Most labs have a website like this where you can really pull up what people actually published in various years. It's very likely that we currently do is somehow grows out or a continuation of these previous projects. I just kind of give you a little bit of a flavor what type of stuff we do. So this is a project where we, it's a very practical thing. PD-LIGO1 is an antibody that's used in the clinic to identify patients who would benefit from immunotherapy. So we looked at how well these assays actually perform in real life. There are multiple different assays. It's a very simple, practical sort of idea. What's the concordance between assays that people use to actually select a particular treatment? If the concordance is poor, then there is a problem with the assay. So this assay actually has a problem with it. So this actually is a similar kind of translational project that looked at the how various markers that we use to select therapies relate to each other. So I'm going to move on to some other projects. So this, again, the same theme of finding immune biomarkers, that is markers that actually define what patient population would respond to immunotherapy and what sort of additional immune targeted therapies would be synergistic based on the expression profile or expression patterns and molecular makeup of human cancers. So this is clinical trial stuff. This might be interesting for you. So this is a project where we actually wondered why people develop cancer at a young age versus at a more typical age of above 60 or above 65. So we looked at germline mutation burden in cancer-related genes. And what this paper describes that Chin, the visiting postdoc from Shanghai, showed is that if you take the space of cancer-relevant genes, and I can get back to this a little bit, at what's a cancer-relevant gene, and you look at high functional impact variants in them, the totality, so that the running sum of these high functional impact germline variants that a particular person is born with is much higher in patients who develop cancer at a young age versus those who develop it late. So the idea is really, really simple. So for a normal cell to turn into cancer, it needs to acquire a particular sort of amount of abnormalities in key regulatory pathways. So it's the running sum of these abnormalities that matters. And when this running sum reaches a threshold, that depends on how much damage you were actually born with and how much you acquire during your lifetime. It's a really neat paper that shows that how this germline variant burden is directly proportional, relates to the age of developing cancer. So we are building on this project now and starting to examine this question that actually what percentage of human genes are relevant for cancer, and could we actually rank them by relevance? So obviously being relevant could be slightly relevant, very relevant. So the very relevant ones we already kind of identified historically in the past like 30 years, but the less relevant ones are actually, it's a pretty gray zone. So that's the project that we are working on. And then how you translate this into something practical in the clinic. So you probably all get it, that if you have a score like this, which is sort of the Cancer Genome Systems Integrity Score, then you could use that score as a risk predictor. And then with this risk predictor score, you could actually tailor risk prevention strategies, screening, prevention drugs for a particular individual. Okay, so I'm gonna show you some other things that kind of just give you a different flavor. So one manuscript that's kind of missing from here actually is another aspect of our lab. I'm not sure why this didn't pick it up. We also do laboratory research and try to identify new therapeutic targets and new treatment strategies. And one of the papers that we actually just published, and I'm not sure why this doesn't show up here, is we looked at how combinatorial screen of existing drugs could actually reveal synergies that we might have overlooked or never been tested in the clinic, and what sort of molecular underpinnings determine the synergy. So we looked at a number of different drugs to find out how they synergize with each other in preclinical cell line models, and what sort of molecular features correspond to these synergies. And another project, and that's the last one that I'm going to mention to you, is try to develop a new antibody drug conjugate, which is interesting mostly for those of you who are more laboratory based. So this involves creating an antibody against a molecule that's highly expressed in cancer, and then trying to optimize the binding ability by shuffling the sequences. It's called affinity maturation, so that you really artificially create an antibody that has a very high binding ability to a particular molecule. And that's a project that one of the postdocs is working on. I'm going to stop here, and if you guys have questions, then I just try to